Hi, so our project is called HCSP, which stands for Homo chloro-sapiens. Chloro meaning green in the Greek language, and so it's about human photosynthesis. I'm Gabby.
I'm Anne.
I'm Claire.
I'm Aditi.
And I'm Kana.
So, our project seeks to produce energy through photosynthesis in the human body, which with an intent to shift humans away from carbohydrate-based food, ultimately reducing our reliance on agriculture and also posing as a very good long-term solution to the global food crisis. We do believe that by moving away from agriculture, an independence from agriculture could really be a catalyst for radical social and economic changes.
So we were sitting down for lunch one day, and I was talking about how I didn't really like eating food and how it always was time-consuming. And then Aditi said, "What if we put chloroplasts in humans?" And I was like, "Wow, that's a great idea." So that's what we decided to do our project about. So we were looking online for animals that use photosynthesis, and we found the spotted salamander, which has algae living inside its cells, and the sea slug, which also eats algae and takes the genes from it to photosynthesize.
And we were also researching ways to carry the chloroplasts to the human skin and not infect them all over the body where they would be pretty much useless. And that's where we got the golden nanoparticles. It's from cancer research.
And the design that we used in the following animations were inspired by the Crash Course videos. Social context. So the changing weather patterns and the rising temperature as a result of global warming are causing agricultural issues. The existing food crisis is turning into a larger problem due to the inability to farm. This is especially impacting countries without any access to imports or humanitarian aid. For example, 50% of people who are facing world hunger in the world in 2016 were Yemeni due to their wasteful agricultural practices and global warming. The food price is increasing due to the crop yield that are impacted by water shortage that is occurring today. By encouraging the provision of energy through human photosynthesis, so we seek to minimize people's reliance on agriculture.
Social impact. Climate changes as a result of global warming are making agriculture more challenging for equatorial areas whose deserts have proven difficult to farm on. Becoming less reliant on agriculture will drive the cost of food down, and prices going down will encourage people to seek out jobs in other fields and save the time and resources committed to agricultural practices. Additionally, moving away from farming will reduce carbon dioxide emissions and water usage in agriculture, saving it for human consumption. Current conventional and organic farming methods for carbohydrate-based foods cover excessive areas of land. And as a result of photosynthesizing, in addition to utilizing current cellular respiration, people will no longer need to consume carbohydrates. With a lower demand for carbohydrates, more land will be freed up to support the growing population and restore natural habitats. Over the last half-century, the world's population has risen from 2.5 billion people in 1950 to about 7.4 billion people today. By 2050, it's predicted that there will be 9.7 billion people inhabiting Earth if the fertility rate does not decrease. Photosynthesis in humans will help the decreasing population growth by eliminating the need for mass reproduction to sustain agricultural workforces.
So to go about this project, we first looked for animals that could photosynthesize like plants do. As I mentioned earlier, we found the sea slug, Elysia chlorotica, which integrates chloroplasts into its own cells by eating algae and taking their genes for photosynthesis. Most importantly, though, we found out that they can live up to 9 months without eating and still maintain normal nutritional rates. After 9 months, they would need to eat more algae to refill on chloroplasts. Similar to the sea slug, our products will only temporarily allow humans to photosynthesize. So some might be glad to know it's not a life-binding decision. We kept searching for other photosynthetic organisms and came across a spotted salamander, the only known vertebrate whose cells share a symbiotic relationship with algae. Chloroplasts were discovered near the mitochondria of the salamander cells, meaning that the mitochondria likely consumed carbohydrates and oxygen created through photosynthesis. In other words, the products of cellular respiration were used in the process of photosynthesis. Human mitochondria produce non-nuclear DNA, or multiple copies of the DNA, unlike that which is contained in a eukaryotic organism cell nucleus. Like our mitochondria, plant chloroplasts also contain DNA. But the problem is that plants have evolved to have nuclear cpDNA. Therefore, it was imperative for us to identify a less evolved plant, or one with non-nuclear DNA. I found the green algal species, Nephroselmis olivacea of the Prasinophyceae class. Several articles wrote about the research conducted to support the idea that this species of algae is a descendant of the earliest diverging green algae. So we plan to have chloroplasts of Nephroselmis olivacea incorporated into our cells.
So now for actually putting the chloroplasts in the human bodies. So first, we took the chloroplasts, which we got from the algae, along with an antibody called HPA012050, which is a lot of numbers. And it'll be attached to gold nanoparticles. And then the antibody will act as a targeting agent, which will guide the nanoparticle to the antibody's corresponding protein, keratin 23, which occurs frequently in skin cells. And then these three components will be combined into a pill form, so it can be digested. And then once swallowed, the gold nanoparticles carrying the chloroplast and the antibodies will travel through the bloodstream to skin cells in the epidermis, which is the outermost layer of the skin. And once there, chloroplasts will become incorporated into the skin cells. And as chloroplasts are hydrophobic, they'll be able to enter the cells through the semi-permeable cell membrane, which only rejects hydrophilic molecules. And then inside the cells, the chloroplasts will begin photosynthesizing, producing simple sugars such as glucose, which will provide a power source for the mitochondria. And these two organelles, the chloroplast and the mitochondria, will act in a self-sustaining loop where the products of photosynthesis power the mitochondria and vice versa, with a byproduct of ATP to power other aspects of the body.
So we have some going further questions. We are going to have to consider if we are ever going to move completely away from agriculture. Also, what would happen to nations who are economically dependent on agriculture? But then we also have to ask the questions of whether or not humans are even ready for these major biological changes to themselves. What will our meals look like without these carbohydrates? And ultimately, how will humans continue to evolve with a changed diet?
So in conclusion, the process of converting humans into photosynthesizing beings involves gold nanoparticles with antibody targeting agents, ferrying the chloroplast to human skin cells, where for months the chloroplast will absorb and convert sunlight into chemical energy. And the production of energy will fuel the existing mitochondria in human bodies, which would then produce the necessary molecules to keep the chloroplast alive, and creating a self-sustaining cycle. And by pushing the human self-sustenance, we would like to encourage a change in our societal behavior, including the reduction of agriculture and the removal of carbohydrates from human diet. And with the implementation of our Homo chloro-sapiens, we hope to rewrite human culture and behavior, making the impossible possible.
And that was us, by the way. Oh, so the last 5 people in that classroom were actually going with us. So this is the crash course version of our cells that we drew yesterday.